Speakers are working.
Do you have his phone number?
I don't. He called me.
I just have to start.
An email.
They thought that this would be our communication for Texas.
Are you texting him? Do you have it? Yeah. Hi, Michael. If you can hear us, just send us a text message or unmute yourself and we can get started.
Hi, Michael. This is Cam Agent. Can you hear us? All right, well, we're starting, so I'm assuming you can hear us. All right, okay, everyone. Thank you for joining us today for Mike Drexler's. Hello. Thank you.
Science!
Yes, let's get started. We're a few minutes late because of a technical problem. Thank you for joining us for Mike Drexler's PhD defense. Mike joined CMS in 2008 for a master's degree with Pam Halleck-Mueller, and he entered the PhD program in 2011 as an MRA student. Prior to that, he did a bachelor's degree at the University of Massachusetts, Dartmouth, and he had experiences as a research fellow at the Smithsonian Marine Station, study abroad program at James Cook University, and he also worked at FWC and NOAA before coming to PhD program. He's currently the lead scientist at the Sustainable Fisheries program at the Ocean Conservancy in St. Pete, and he works closely with NOAA and the Gulf and Pacific Fisheries Management Councils and SSCs. He is responsible for providing scientific support to the Ocean Conservancy. For example, he helped develop the Poseidon model, which is an agent-based approach to capture human interactions with the marine ecosystem. Mike is a very impressive publication record for someone at the beginning of his career. He has 9 peer-reviewed publications, including 2 first authored and an additional 3 now in review. That's pretty good, 12 for his PhD. In addition, he has 4 technical publications. He has presented his research in Latvia, France, Spain, Norway, and the US. And he has been recognized with a Gulf Oceanographic Charitable Trust Fellowship, a Student Participant Scholarship for Travel, a Link Foundation Graduate Fellowship, and an award for excellence in lab work from FWRI. He is also more recently a co-PI on a Marfin project. In terms of service, he is a founding board member of the Tampa Bay Waterkeepers, a board member for the Hands for Hope nonprofit organization, and he has served as a marine educator for Nature's Classroom, as well as a student representative in the CMS Faculty Evaluation Oversight Committee. His committee is composed of myself, Dr. Cameron Ainsworth, Dr. Ernst Peebles, Dr. Frank Mueller-Carger, and Dr. Steve Borowski, who you will know. And joining us via the internet is Dr. Mike Sharippa from the Southeast Fisheries Science Center at NOAA. Michael is adjunct faculty at USF and UMiami, and he's been involved with the Integrated Ecosystem Assessment Program, as well as the Fisheries and the Environment Program, and has been a strong advocate for ecosystem-based management and modeling in the Gulf of Mexico. The defense chair is Dr. Rob Ellis. He is an associate research scientist at the Florida Wildlife Commission. And Dr. Ellis will now tell us just a little bit about the format for the presentation today.
Sure. So in a minute, Mike's going to go ahead and give his presentation about his defense. That should take about 50 minutes. After which we'll open up to the floor for questions. The committee is asked to participate if they'd like. Just keep it to general questions. and hold your more specific things. After the question period, we'll take a short break, at which point we'll reconvene. The public is welcome to join at that point, but we're going to limit questions to the committee members until they're satisfied that Mike has defended his dissertation. So I think that's it from me.
Okay, thank you all for coming today. Very appreciate it. So I'm going to talk about my dissertation, evaluating the use of larval connectivity information in fisheries models and management in the Gulf of Mexico. And this work was funded initially by NOAA and USF and many in-kind hours by the Ocean Conservancy. And so briefly I'll talk about how is used or not used in to attempt information on ecosystem context. We'll run some ecosystem simulations with and so jumping right in, fish in fisheries management. First answer is it's not really. To a single all fish in nature, that information's incorporated some in different ways, and so just some examples of spatial state-specific landings, in this case red snapper, and scientific surveys such as the CMAP trawl survey. But in fisheries management, all of this spatial information is collapsed down to a management unit. And these management units are typically defined via genetic connectivity, and in the Gulf of Mexico, for the most part, span the entire Gulf of Mexico. And so determine the management unit, and then collapse all of this information down to sort of dimensionless time series to provide information for our unit stock, develop stock assessments, and provide critical management reference points, such as do we have enough fish in the water and are fishing rates sustainable. And so for this stock here, the goal is to keep your stock in the green, where biomass levels are high and able to replace themselves and fishing rates are relatively low. We can also use spatial information in sort of active closed area management. And so there's two general types of closed area management we can use in fisheries management and marine ecosystems. The first being to protect important habitats, such as coral areas, all types of essential fish habitat, and spawning aggregations are three examples of closed area management used to protect habitats. And then the second, more complicated piece is to increase fisheries yield. And the idea behind using closed areas to improve fisheries yield is that you protect a certain portion of the stock from fishing, allow that portion to grow, reproduce, and have those impacts, be it larvae or actual movement individuals, spill over into areas that can be exploited for fisheries management. But in all cases, what we find is when evaluating closed areas for any scenario, you really need a case-by-case understanding of the spatial structure of the stock and species and fisheries being impacted. And why is this? Well, population connectivity is a difficult beast, and there are a number of contributing processes that contribute to total population connectivity. And so on the x-axis, you can see some example of what these processes are, and the y-axis is a sort of conceptual representation of the spatial context of each of these processes. So generally, population connectivity can be defined as where are fish spawning, where do their larvae or spawn disperse to, and then post-settlement effects such as predator-prey interactions, habitat availability, condition. And so in this dissertation, we'll attempt to address each of these using models. And the first step, so where are the fish? We'll develop some species distribution models using the data we have available to us. Second, if we know where the fish are, we can simulate where the larvae disperse to, so we'll implement some larvae dispersal models. And last, can we incorporate all of this information, the spatial information and simulation, in a meaningful way to provide management type advice? And so to do this, you need more models, but ecosystem models to bring all of this connectivity information and simulate the ecosystem impacts of that connectivity to try and understand how important is it that we include this information in our management advice and models. And so jumping right in, species distributions models. This work was published back in 2013, generalized additive models used to predict species abundance in the Gulf of Mexico, an ecosystem modeling tool. And the goal was to address this first step of population connectivity here. Where are the fish? Where are they spawning? And so the first step to implementing any spatial ecosystem model is, where do you put all the fish? So it might seem like a straightforward question. When you dig into it, it's not so straightforward. And so how many of our space fish live in our Atlantis ecosystem box up here, or one of these Osmos, which is another multi-species ecosystem model, grids in the West Florida Shelf. Furthermore, how do you do that in a multi-species context for a whole slew of species that you're modeling in the ecosystem? And furthermore, can we use the information we have, say in the northern Gulf of Mexico, to make some informed estimates of where the fish might be in the southern Gulf of Mexico, or more generally, areas where we don't have direct observations? And so we took the approach using models. I used a generalized additive model approach. But the general idea is to use the abundance information we have, which is generally available in the northern Gulf of Mexico, and so you can see some example measures of relative abundance. Link it to information we do have across the entire Gulf of Mexico. So we do have environmental data across the whole Gulf of Mexico. And if we can determine reasonable relationships between these two things, we can create prediction maps for across the whole Gulf of Mexico. And so the abundance observations I'm using are the CMAP groundfish survey, CMAP groundfish survey. This is a trawl survey run by the Gulf States Marine Fisheries Commission and FWRI, another state research agencies. Summer and fall survey, they go out every year. Do this roughly summer and fall, depending on sampling, and stratified by statistical zones and depth. So generally you get a representative sample, trawl sample of what's out there across multiple, at least across the U.S. sort of area. And the environmental data I'm using, I use 5 environmental data layers here. These are climate scale environmental maps. And this information was, so this is multi-species averages across the whole Gulf of Mexico. Most of this information can be found in the NODC, Gulf of Mexico Regional Climatology. Sediment data can be found from the U.S. seabed database. But generally, all of this information comes in different resolutions. So everything was interpolated to a 0.1 degree grid cell using a variety of methods to create these continuous maps to both fit models and make predictions. And so what are generalized additive models? Well, they're a direct extension of generalized linear models. Sort of think of them as fitting, uncooked spaghetti or straight lines to your data, as opposed to GAMs, which give you generalized additive models, which give you a little bit of flexibility, sort of like cooking, using cooked spaghetti. And instead of fitting, straight lines or a predetermined shape of a curve, you can actually just fit a smoothing function to your data, like a moving average. And so to fit these models, we incorporate these 5 environmental layers, and the equation here, where I guess this is eta, represents the expected abundance at a given grid cell. Gamma is a link function, in this case negative binomial, and S is the smoother function I talked about. You sort of cook spaghetti. And the smoother function I use in this case is a thin regression spine fit using penalized likelihood estimation. And important in this package, this is fit using the MGCV package in R, as most is it allows you to apply this extra penalty for weak relationships. And so when the fit is poor to any one of these layers, you can apply this extra penalty to the fit and essentially remove it from the model, at least the impact to your predicted abundance. And so this is really useful in sort of the semi-automation when you're doing this for multiple species in the Gulf of Mexico. And so I'll walk through an example of pink shrimp, as we do in the paper. And so you can see the CMAP observations of pink shrimp here, where dark areas are areas of high observed CPUE, or relative abundance. And you can see the spatial distribution of that data. And you can couple this with anecdotal data from a species profile report way back from 1982, where they identify pink shrimp maximum densities along the Florida and Yucatan coasts, where we actually don't have data in here. And you can see a conceptual sort of drawing they include, which are the pink shrimp hotspots off of Florida historically, of both the species in the fishery. And so we can fit those abundance observations to those environmental rasters I showed you. And so looking on these diagrams along the y-axis, you see the additive effect when you're predicting using these models to the predicted pink shrimp abundance, given a set of parameter values for any of these variables. So what we find is pink shrimp tend to occur in shallow depths, at moderate temperatures, across a range of surface chlorophyll A, probably not the best predictor of benthic species, and a really poor fit to oxygen. And last, a preference for the carbonate sediments of the West Florida Shelf and Campeche Bank, as opposed to mud sediments where you would expect to find white and brown shrimp. I'm so quite happy with at least the qualitative results. And so what do the prediction maps look like? You can see the prediction of relative abundance across the Gulf of Mexico. I see a strong band of pink shrimp in those depth areas that we identified in both the Campeche Bank and dry Tortugas, again, where we didn't have data, abundance, direct observations of abundance. And so we translate that data into the Atlantis polygons that we'll be using for ecosystem simulation. And so this is just collating this into these polygons, simple average. And what we find is, that band of high pink shrimp abundance along the West Florida Shelf with relative certainty. These white areas here indicate, we're predicting these areas with relative certainty. And relative uncertainty on sort of the extremes of the environmental data that we have. And so looking at the diagnostics, so it's qualitatively, the maps look good. Looking at the diagnostics, when we initially fit these models, you reserve a third of your data to evaluate your model, and so you fit your model using the bulk of the data. You use those predictions to evaluate your model, and the remaining third that wasn't used to fit the model. And so these are the results. If the model is perfect, you expect a one-to-one line going through your system, where your observations directly line up with your predictions of relative abundance. And so looking at the point estimates, those individual grid values, the fit isn't actually that great. You can see the problem of sort of zero inflation here, where your zero observations span your full range of observed CPUE. But in general, you do get contrast between areas of high and low abundance. And so looking at when we aggregate this to the Atlantis polygon level, you see an increase in that contrast, closer to a one-to-one relationship. But again, in both of these scenarios, this is really not a really good model, especially predicting zero abundances. But we do get that important contrast. And it's important to remember the context that we're using this information. This is for a first guess in our ecosystem simulation model. As soon as day 2 clicks over, all of the ecosystem dynamics come into play. And so in the ecosystem model context, this is really just a first guess, Bayesian prior sort of. But in the end of the day, we really calibrate our models to our ecological dynamics in the model and input this as a sort of a first guess. So overall, we find the gamma provides sufficient information for initializing ecosystem model. It gives us that contrast. Doesn't give us a really good picture on the absolute abundance of these species. Okay for relative abundance. Limited predictive power at zero abundance. We're really having a hard time fitting those zeros. But it does represent a data limited and data pragmatic approach. We're using models to try and make informed estimates from data we do have into areas that we don't have any. And so this is really useful for that, especially these extrapolations to the southern Gulf of Mexico. And we find spatial aggregation to these Atlantic polygons. It improves the fit and is the resolution that we'll be using this information. So additional considerations, why didn't I use, and of course we apply this to, I think, 40 groups that we capture in the CMAP database, and we applied our functional groups in the Atlantis model. So why didn't I include a latitude variable? Well, it's really not suitable for extrapolation. So we don't have any data in the southern Gulf of Mexico. So I don't have any latitudes to predict from. Ultimately, future iterations of this work, which I'll talk about later, uses a delta gamma approach. And this approach basically models presence-absence data and measures of abundance where shrimp or any species are present separately. So it gives you some additional flexibility a more intuitive interpretation of presence-absence data and what's going on with those zero values. It also provides an additional flexible variable selection. So you can use different variables to fit your presence-absence model and your abundance model. I didn't evaluate any interaction terms, and spatial autocorrelation isn't dealt with. And I'll, again, talk about all these later at the end. So jumping in, where are the fish? We've demonstrated where pink shrimp are. We can turn this crank for a whole bunch of species in our ecosystem model. And so larval sink source dynamics can inform spatial management, a Gulf of Mexico case study. So we've identified where all the fish are, at least made an educated guess. So where do they go? We can do this using dispersal models. So looking at the larval fish information in the Gulf of Mexico, the information is quite limited. So here's an example of a tuna study, which uses the CMAP ichthyoplankton survey, which goes out every year, does plankton tows, and identifies and quantifies the larvae samples. And the resolution is actually quite good for tuna species in the offshore areas. But species resolution is actually quite low for reef fish species in the Gulf of Mexico. And so this is an example of red snapper, studied by Manny Karnauskas, commercially important species in the Gulf of Mexico. And she had to resort to doing sort of sensitivity analysis. She had to make some assumptions about where the larvae are in the water column, because we don't actually have good information on where they reside for key species in the Gulf of Mexico. So overall, we don't have a ton of information about where the larvae are, at least to create an accurate picture about where all of our species are dispersing to across the whole Gulf of Mexico. So it's just me. It's sort of a fruitless effort to try and nail this for all of the species in the Gulf of Mexico to make it super realistic. So we decided to take a sensitivity approach. So how important is it when we make some simplifying assumptions about how our larvae disperse that we include this information or not? And so to do this, I created a simple larval dispersal model. It uses the Gulf of Mexico HICOM daily velocities. This is experiment 20.1, which runs from 2003 to 2009, and provides velocity vectors on a 1/25th degree resolution, across the whole stratified water column. And you can see an example of the resolution. There's about 25,000 nodes that provide surface velocity vectors, and basically the dispersal model I created incorporates all of this information, daily velocities, and integrates, estimates the velocity from the 3 surface layers. So in this case, we're assuming we made a simplifying assumption of surface transportation for all of our species in the Gulf of Mexico. We know this isn't right for all of the species. Dr. Peebles and Dr. Weisberg have a paper demonstrating in the case of gag grouper, they think that gag utilize a bottom transport pathway. But again, for sensitivity purposes, realized we weren't going to get it all right, given the information we had. And so we can also use that information and create a spawning calendar of all of the species and species groups that we're modeling in our ecosystem. So this is just a calendar with the day of the year along the x-axis and some example species. I haven't listed them all along the y-axis. And on the left side of the bar, you can see the date we're assuming they spawn, and the length of the bar is their pelagic larval duration. And so I'll walk through an example of white shrimp demonstrated here, which spawn in the summer and have a larval duration of around 30 days in this model. So here are some example trajectories. So the northern Gulf of Mexico and southern Louisiana and Texas is sort of the white shrimp center of abundance and also center of the fishery. And so I'm just here just demonstrating the particle trajectories of these key 4 Gulf of Mexico boxes, the trajectory of those particles across all of the years. And what we see is particles being following a number of major hydrological features in the Gulf of Mexico. Eddy's moving to the east, loop current. And on an annual basis, we see different strengths of particles moving east to Texas and west west to Texas and to the east of the Mississippi River from year to year. And important, all of these particles that we're tracking actually carry the proportional abundance of spawners. So you can see our sort of map of relative abundance here. Each of these particles are carrying a weight of the proportional abundance of the spawners. So what we're essentially saying is for each of these boxes, the proportional abundance in each of these grid cells is correlated to the number of eggs or larvae that those grid cells will be producing. And so we weight each of these particles according to essentially the each of these grids, carry that information, and we can sum those proportions up to understand the level of connectivity, say between this northern Gulf of Mexico box here to a number of other boxes. And so looking specifically at this large box here, which is the sort of central area for white shrimp, what we find through these simulations is white shrimp is mainly self-recruiting, this large red dot here, and while the annual strength of particle movement varies from to the west or east depending on the given year, on average they seem to be contributing, this box seemed to be contributing larvae in roughly equal proportions on average, if you average this over multiple years. So you can take your long-term average, estimate what average recruitment might be. So the dark areas are average recruitment on both our fine gridded system and Atlantis polygons. And flipping this on its head, we can also ask questions: where are your larvae coming from? So in this plot, I'm asking how far are the larvae traveling? What's the displacement distance of those larvae or particles that are arriving in that northern Gulf of Mexico box? And so in most years, the displacement distance is actually quite low, and we identified most of the recruitment is actually self-recruitment, so that's not surprising. But what is interesting in these anomalous years where particles are drifting from up to 400 kilometers away, in this case 200 kilometers away. And why this is interesting, I think, is because it's these sorts of anomalous years that define genetic connectivity over the long term, but it's these types of years that are important, these average years that are important for fisheries management, where you're setting annual quotas that are strongly dependent on annual recruitment stream. And so we've developed maps of where we think fish or species or shrimp are. We've developed maps of where they think they end up. Both of those use a measure of proportional abundance in the Gulf of Mexico. And those are scaled by absolute abundance in the model, but ultimately the units we're using is proportional abundance across the whole Gulf of Mexico. So we can simply overlay those two maps and subtract them and develop an index of sources and sinks. So in this model, I'm defining sinks as a grid cell or polygon that receive more weighted particles than they produce. And a source produces more of these weighted particles than they receive. So you can see the resulting pink shrimp map up here, where inshore areas tend to be sinks of white shrimp for white shrimp, and the strong bar of source area of white shrimp in the northern Gulf of Mexico, which is generally representative of white shrimp life history. Unfortunately, that white strong source signal gets diluted in our Atlantis polygons here. So we're always dealing with this issue of resolution, but I'm just showing this as an example. So key findings from these dispersal scenarios. In the case of white shrimp, we see a high degree of self-recruitment of white shrimp if you assume a surface dispersal pathway. Source and sink patterns are driven by a spatial interaction of settlement and biomass. So when we create those maps and overlay them, we see it's not driven by either where the shrimp are or where the larvae are entering, but it's really an interaction of the two. And three key products. One is a spawning calendar. Two is a measure of larval connectivity between any given box and all of our boxes. And three is these source sink maps. And so chapter 4, evaluating the use of larval source sink dynamics in a management context. Closed area management evaluation using the Atlantis ecosystem model. So far, we've created species distribution models to try and understand where spawning output is occurring. We've created dispersal models to disperse our larvae, at least to understand the importance of dispersal in these simulations. And so now we're going to try and bring all this information together in an ecosystem simulation model, specifically Atlantis, to one, represent these post-settlement effects, such as predator-prey influence and habitat, and also as a simulation tool to try and develop management advice. And I'll walk through a case study of reef fish management in this chapter. So this is just a plot of the annual catch limits of important species in the Gulf of Mexico. You see snappers, groupers, and amberjack and triggerfish as reef fish here. And we'll incorporate all of this information using the Atlantis ecosystem model. So even though I spent this ungodly amount of time working with Atlantis, I'm going to say very little about it in this talk. And just important to know that it's a end-to-end ecosystem model and incorporates everything from the oceanography and lower trophic levels to the higher food web fish and fisheries. And so there are a number of models that do all of these things well and probably better, but there are very few models that incorporate all of this information together. And so key to this chapter is that at the heart of the model, these ecology submodels captures key ecological processes for all of the species we're modeling in the Gulf of Mexico, and specifically predator-prey interactions defined by a diet matrix and diet data. And the model is spatially explicit, also important for this talk. And it's spatially explicit in 3 dimensions. We broke the Gulf of Mexico into 66 polygons that roughly delineate major biogeographical features, such as country EEZs, shrimp grids, depth contours, etc. And there are 90 functional groups in the model, 48 of which are fish. I'll be talking about 10 reef fish functional groups. And a functional group is simply individual species or a group of like species or gills. But the one process that is relevant to this talk is recruitment. And so in this model for our age-structured species and fish, we use a Beverton-Holt stock recruitment relationship, sort of foundational piece of fishery science, but generally it defines the number of recruits you expect for a given size of spawners. And all that this shape of this curve says is that number of recruit per unit spawner will be high when resources aren't limiting, and as they approach carrying capacity and resources become limited, you produce less recruits per unit spawner. And so the total number of recruits in the system are estimated using this relationship. They're then further distributed according to where we think our spawning biomass in the system is, relatively across these Atlantis polygons, and that's typically it for Atlantis ecosystem models where they end. So most Atlantis ecosystem models use the assumption of self-recruitment. So where their recruits are produced, they also end up, basically inferring most of their life cycle is occurring within that same individual box, aside from some adult movement. Also not a great assumption. So we're adding the extra step of defining the connectivity matrix. So how many recruits does the total system produce? Where were they produced? And then how are they dispersed? And we can do this via a simple connectivity matrix in Excel that defines for a given source box, so this red area here, what is the proportional contribution to each of our destination boxes? We can define this for all the boxes in the model. So that's how it's incorporated into Atlantis. And when you incorporate this information, previously Atlantis didn't have dispersal, so the dynamics changed quite a bit. So the model was retuned to represent similar biomass and catch levels after 10 years of simulation. Also structure of the stability of the age structure of all of our functional groups in the model, and persistence of key species throughout the simulation. So the tuned model is then taken and run with some 3 general groups of scenarios with the tuned model. So the first scenario is the status quo. So status quo represents the current day picture, actually 2010, close to current day, picture of closed areas in the Gulf of Mexico. And so this map is a little bit misrepresentative of the areas that are actually closed in the Gulf of Mexico. And the important thing here is that the status quo uses highly precise closed areas, sometimes over large areas which are generally open or closed for a very short amount of time, and targeting precise fisheries, so they're not closed to all fishing, aside from small areas in the Gulf of Mexico. But I created a picture of what the status quo closed area scenarios look like in the Gulf of Mexico. Seasonal closures switch on and off in the model depending on season. All of those things are captured in the status quo scenario. We also implement some generic scenarios. We randomly close 10, 25, 50, and 100% of the boxes in the model. Of course, this is totally unreasonable. It would never happen. Pretty dumb way to go about management, but it serves as a nice benchmark to compare our results. And the last and most interesting set of scenarios is the source sink scenarios. So as you recall, we developed a number of source sink index maps for all of the species. And so I simply asked, what are the most important areas that are acting as reef fish sources and sinks in the Gulf of Mexico? So I simply took all of our reef fish functional groups in the model. These are summed up their source sink maps, ranked their importance on my source sink index scale, and then closed the 3 and 10 most important source and sink boxes for the aggregate reef fish sort of group in the model. So what do the results look like? Well, everything is compared to the status quo. So on the plot, you'll see the changes in biomass and catch relative to the status quo, our 0 value here, as we increase the area of the size of the closed area from 0 to 100%. And so in the case of the generic closures, what we see as we increase the size of the closed area along the x-axis is an increase in biomass up to around a 50% closure in the Gulf of Mexico, consistent decrease in catch, biomass levels off, catch continues to decline. So this to me indicates a potential reef fish the reef fish are approaching their carrying capacity at around a 50% closure in the Gulf of Mexico, and the point where these cross is a potential sort of Pareto efficient solution where you get a win-win. You get some additional biomass and some additional catch relative to the status quo. Of course, we're not about to randomly close 50% of the Gulf of Mexico, but it serves as a nice baseline to evaluate these source and sink closures. So what do the sink closures look like? So you can see the effect as we move from a 3-box closure, which is a small area, to a 10-box sink closure, which is a larger area on our x-axis here. And what we see is catch in red and biomass in blue more quickly converge to the status quo line here, suggesting that targeting your closures, targeting ecologically significant areas, you actually get the value of a really large, ill-informed closed area by closing a much smaller portion of the area. You can achieve status quo benefits by closing around 15% of Gulf of Mexico. Gulf of Mexico, and moving from a 3 to 10 box closure reduces catch by 45%, and you make some gains in biomass. Now looking at the source closures in triangles here, you see I add them to the same plot. What you see is as you move from a 3-box source closure to a 10-box source closure, you make similar gains in biomass, and while reducing catch by a smaller amount. So instead of a 43% reduction in catch, we see a 20% reduction in catch. That intercept well above our line here, again suggesting this may be sort of a win-win scenario where we're able to target ecologically significant areas, specifically sources, to make some additional gains in biomass and catch of the aggregate of the reef fish stocks. Stocks and overall, again, targeting these important areas, you get much more bang for your buck than using these uninformed closures. It's important to note the absolute changes we're looking at on this plot is actually quite small. So increase catch of 1,000 metric tons for all of your reef fish stocks in the Gulf of Mexico isn't a huge gain, but it is a gain. I'll talk more about that in a second. So digging one layer deeper, what's going on here? What's driving these changes? Well, what we find is the groupers and small reef fish are most sensitive to these source and sink closures. What we find is groupers are extremely sensitive to catch when you start closing areas, and small reef fish biomass are extremely sensitive to either of these areas. And so when we see the gains in biomass and losses in catch, generally what we're looking at is gains or losses in catch of red groupers, and biomass of small reef fish. But when we compare the source closures in purple, this is the 3 and 10 box closure, to the sinks, what we see is as we move from a 3 box source closure to a 10 box source closure, it has a very small impact on grouper catch compared to the sink closure, and large impacts on small reef fish biomass. And why is this? Well, unsurprisingly, the location of the closures matters a lot. So on the plot here, you can see a this is a plot of red grouper catch, where purple is no catch and red is sort of your hotspot of catch. And what we find is, here's the status quo. You can see sort of the hotspot of red grouper fishing on the top there. What we find is, I didn't specifically choose where these closures were located. I simply did that ranking and closed the most important sinks. Well, the most important sinks all happen to occur along the West Florida shelf here. So when we close these sinks, there are huge implications for targeted species like red grouper that are caught here, and has large impacts on the catch, as opposed to these source closures, which are closing boxes along the shelf of the West Florida shelf and south of Texas. So the impact of red grouper catch is actually much smaller. In closing the offshore boxes, you actually make pretty large gains in small reef fish biomass, which are not economically valuable but provide ecological services. And from a value perspective, if you're a fisherman, there's likely increased value in not closing these inshore boxes when you're going out fishing, just because of the distance traveled. So conclusions. We find protecting source closures is going to give you your best bang for your buck. And essentially what you're doing is protecting those offshore closures and creating that spillover effect for fishermen to exploit fish inshore, as we semi-expect. So in some cases, a validation of the model. We find a clear convex relationship between reef fish biomass and catch. So you're able to see, aggregating across reef fish, that carrying capacity for the entire reef fish stock. But ultimately, any of these outcomes are going to be highly dependent on management goals. I was looking at a single goal, increasing the aggregate reef fish biomass and catch. But in reality, any of these goals are going to be much more highly precise than what we're doing here. But what this does offer is an operating model to specifically apply that case-by-case understanding of spatial structures. So you can start to evaluate these highly precise management goals and simulations. And I also mentioned that the overall gains, the absolute gains in catch and biomass, were actually quite small. And initially I was surprised by this, but in reality, the Gulf of Mexico and U.S. fisheries are generally well managed. We have targets. We try to manage them to achieve maximum sustainable yield and biomass at MSY. So you actually wouldn't expect the gains in any of these scenarios to be extremely large, although the economic benefits could provide some additional value. And so conclusions and recommendations. So we've attempted to incorporate all of these processes of population connectivity using models and spatial information in the Gulf, using the data we have, not always the data we want. But there's one key missing piece of information. As I mentioned before, the key to predicting where larvae are going to disperse to is understanding their behavior. And so most of you know larvae can migrate vertically in the water column and exploit different pathways to disperse themselves across the Gulf of Mexico. We don't really have a solid understanding about how these larvae are moving, at least well at a high species resolution across a lot of species. So I think the key to making these models realistic and tactical in nature is really understanding this larval behavior process, understanding the pathways larvae are exploiting to get to where they go. And there are some examples and people working on this information. So Kelly Bender and Cam Ainsworth Lab is specifically looking at the ichthyoplankton data we do have to try and understand if you can create these sort of vertical profiles of where larvae reside. I showed you an example of Mandy Karnauskas' work using the connectivity modeling system, which is sort of a generalized dispersal tool that can incorporate behavior information. And taking a sensitivity approach to try and understanding how sensitive our goals are. This is a measure of relative recruitment strength for red snapper if you assume a whole range of movement pathways. There's two examples of this that we can expand upon and start to incorporate into these types of operating models. And the second is improving these species distributions. That initial paper was a pretty crude and quick guess at where the species are. Perfectly suitable for our use in Atlantis ecosystem models, but other types of ecosystem models that are being developed in the Gulf of Mexico, specifically osmos, is actually highly dependent on species distributions, and the overlap of predator-prey is actually used to define who's eating who, the predator-prey interactions in the model. So require a much more precise distribution of species. And so our node and many others have been leading and are wrapping up a project now to improve the methods we're using and incorporate a wider range of information. So we have compiled 37 monitoring databases, 21 environmental parameters after considering many, many more, into a large database that's now available via the Restore, I forget the name of it, I'm sorry, but the Restore Council database. I can point you to that. Happy to point you to that because it's a really valuable source of information. Improve the binomial gamma approach, deal with spatial autocorrelation using geostatistical models, improve our resolution across life stages, and improve variable selection process. And this is all published. I'm happy to point you all to that. And so thank you for everyone for your patience and support through the years. Thank you to the other Dr. Drexler kids and everyone that's helped me along the way. And again, sorry I've taken so long, but as Kent showed you, I promise I've been busy and productive, so thank you for your patience. And bring on the Space Force. Any questions?